everyone. My name is Alexandra Adams, and I'm a chemical engineering PhD candidate at Stanford University. I first became really interested in research during my undergraduate education, also in chemical engineering, at Miami University of Ohio. I'm really excited to share with you today some of the research I've been working on during my PhD. So, my PhD has focused on harnessing the power of recognition in DNA for biosensing. I know that's a lot of words that don't necessarily seem like they should go together. So I work on molecules called DNA aptamers, and these aptamers work as synthetic antibodies. So you can think of your antibodies that maybe are present in your COVID-19 test, where they're able to capture COVID molecules and give you that pink line whether or not you have COVID. There's another class of molecules called DNA aptamers that can perform the same functions but are much easier to make, found through a process called selects, where we're able to use directed evolution approaches to find sequences that bind to our specific target molecules, whether that be proteins, other nucleic acids, or small molecules. And this actually allows DNA aptamers to bind more things than your antibodies might be able to. In biosensing, this is really important and really awesome because it allows us to capture target molecules in really complex samples. So if you're looking for one or two molecules in a drop of blood, you're able to find them with your DNA aptamers. There's a special class of DNA aptamers used for biosensing and diagnostics that are called split aptamers. Usually, we have one continuous strand of DNA. But in a split aptamer, we're actually able to cut this into two pieces. And you're able to tag these pieces with a fluorophore and a quencher, basically just saying you're able to tag them with something that lights up and something that doesn't light up. And when you introduce your target of interest back into the sample, you bring the light up part near the part that stays dark and you lose all of your signal. The problem is that there's kind of a lack of general strategies for identifying where we should cut our aptamers so that we're able to get really good modulation in fluorescence levels with and without the target present. Our lab has a unique platform that is able to actually screen for millions of these sequences all at one time and tell us how they're behaving in the samples. We're then able to use computational methods and different analysis tools to identify unique sites for splitting our aptamers so that we can get better performing sensors. That ends with us getting this really big data set that we parse through and identify locations that either result in small changes creating better binders or worse binders. And ultimately, we were able to show that we could utilize our split site locations to get better performing sensors for detection of SARS-CoV-2 RNA. And so with that, I would really like to thank the ARCS Foundation for this scholarship and the ability to pursue my research and continue exploring the world of biosensing. Thank you.